Everyone, as you file in, go ahead and start working on this warm-up problem. This is going to test our understanding of what's going on with metabolic activity in the arterioles, or sorry, in the tissues versus constriction and dilation of the arterioles. So basically here, I'm asking you to think about active hyperemia, and then I want you to think about what would happen in the opposite situation. Give you a second to work on this before we go over it as a group. Okay, I'm going to pull up a whiteboard and start making my own sketch, but continue working on yours. Okay. Could someone give me a thumbs up if you see my whiteboard? Awesome. Okay. So what we're thinking about here is we're thinking about a tissue that is becoming less metabolically active. Oh, still got people joining us. Okay. So I'm going to start out by drawing our tissue and our arteriole. We'll draw them in the same colors as the textbook drew our opposite example. So here's going to be my tissue. We've got some extracellular fluid. So we've got this space here. Then we have the arteriole running past it. Okay. So the first question we want to ask ourselves, because we know that the arterioles are responding to the concentration of oxygen and carbon dioxide here in this extracellular fluid, this space between the tissue and blood flow. So we want to ask ourselves, what's happening to those concentrations regarding this tissue? And I'll try to keep our colors the same as the ones the textbook used too. So we're going to be thinking about our O2 is purple, and our CO2 is going to be green. Okay. So in a normal state, these two are in equilibrium. O2 is getting used up as fast as it gets from the blood, and then the CO2 is being washed away as fast as it is produced. When this tissue becomes less metabolically active, that really means, right, is it's not doing as much cellular respiration, it's not breaking down as much glucose, which means it's not using as much O2. So what we're going to see is we're going to start to have a buildup, right, of O2 here. Right. So conversely, we're having this buildup of O2. We're also going to have a change to the CO2 concentrations, right? Imagine we had some CO2 here to begin with, but we were having an even flow in from the tissues and then out from the arteriole. But now that this tissue is less metabolically active, it's not producing as much CO2, right? So we're going to have very little CO2 here. So we got lots of oxygen, not very much CO2. So this is like the opposite situation compared to our active hyperemia, right? In our active hyperemia, we didn't have enough oxygen and we had too much CO2. So we needed to make this vessel bigger so that we could wash away that CO2 and get oxygen here faster. But what this situation is telling us in this extracellular fluid is that we have too much blood flow compared to the amount of oxygen being used by our tissue. So what we're going to see here, instead of vasodilation, right, I'll use a darker red for the second state, right? What we're going to see here is we're going to want vasoconstriction, right? We're going to get a narrower arteriole because we do want to maintain the concentration of the oxygen and CO2 in equilibrium here in the extracellular fluid. That's really what that's the big takeaway of our intrinsic control of the blood flow, of this local control, this response to O2 and CO2, is it's all about trying to maintain this constant steady state of O2 and CO2 here. Okay, so in this situation, we would see vasoconstriction, which means that the arterioles would decrease in their diameter, decrease in their radius, which means we're increasing the resistance, right? When we increase the resistance by decreasing the radius, we get less flow to this tissue because it doesn't need as much flow because it's not using as much oxygen. Okay. Questions about that before we move forward for our remaining material on arterioles for today? Okay. Drawings, and we're going to share PowerPoint again. So that's what we were talking about just now. Okay. So another quick sort of way of thinking about our hyperemia, active or reactive, we're going to run through one of these flow charts that our book likes so much, showing us the feedback loops and the changes in each of these different factors. Okay, so one way we can think about these things and understand them is by drawing them out. Another way we can think about these things and trace these relationships is by drawing flow charts. We like to present multiple ways, right? Because people think about things in multiple different ways. Okay, so here we're doing a comparison between active hyperemia, which remember is when we're talking about the metabolic activity of the tissue, and specifically when it's becoming more active, right? So it's doing more cellular respiration, breaking down more glucose, needs more oxygen. We're comparing that to reactive hyperemia, which is the situation where we're increasing blood flow, but due to a blockage beforehand. So this is when we have more blood flow because blood flow has been cut off for a period of time. So we can see that they have different root causes, right? So in our active hyperemia, we're starting out with an increase in the metabolic rate, whereas in our reactive hyperemia, we're starting off with a blockage, which we can think of as a decrease in blood flow. So for active hyperemia, that increase in metabolic rate directly increases oxygen consumption, which means that the tissues, right, are soaking up more oxygen. And it increases CO2 production, which means they're releasing more CO2 into that extracellular fluid, into that space between the tissue and the blood vessel. Okay. So here's where we see what's happening in that extracellular fluid, that fluid that's abutting the arterioles, that's right next to the arterioles. So these are the concentrations that have the direct effect on vasodilation versus vasoconstriction. And we can see that both of these situations, an increase in metabolic rate or a decrease in blood flow due to a blockage, have the same effect on that concentration between the tissue and the arteriole, right? They both decrease oxygen concentration. In the case of active hyperemia, because more oxygen is getting used up. In the case of reactive hyperemia, because we just don't have enough oxygen getting there because we don't have enough blood flow, right? So we have like old stale blood kind of sitting around. We've already used up all the oxygen. Okay. We have an increase in CO2 concentration here because of this increased metabolic rate. And here, because of the decrease in blood flow in reactive hyperemia, we don't have the blood to sweep away carbon dioxide. So those tissues, they're still metabolically active. They haven't changed their metabolic activity, but we don't have blood flow to sweep away that carbon dioxide, so it's building up. Okay. So both of these situations have the same effect on the local arteriolar smooth muscle. And remember that the arteriole, right, is the vessel carrying the blood to the capillary beds. And it's regulated by the smooth muscle that surrounds it, right? Arterioles have a layer of smooth muscle that either contracts to constrict or relaxes to dilate. So we have the same concentration, so it has the same effect on smooth muscle. Smooth muscle doesn't directly know anything about the metabolic rate. It doesn't directly know anything about the blood flow. What it's responding to is these changes in the O2 and really the CO2 concentration. Okay, so both of these situations, CO2 went up, O2 went down, has the same effect on the smooth muscle because that's the same change to concentration. So both of these situations, we're going to have vasodilation. So the smooth muscle is relaxing, meaning our radius of that arteriole is getting bigger, which means our resistance is going down. We remember we have that equation of flow is equivalent to change in pressure divided by the resistance. Okay. So here we see our resistance going down, right? And the resistance on that equation is the divisor. So we might say flow is inversely proportional to resistance, okay? Which means that the blood flow is going up. Inversely proportional means they're doing the opposite thing, right? As resistance goes down, blood flow goes up, which means we increase the delivery of oxygen and we increase the removal of CO2. So we're basically fixing this change set up by our initial problem, right? Okay. So then we have our final state here. Once we've made this fix, we do have a negative feedback loop, right? Because as we increased O2 delivery, we increased the O2 concentration. As we increase the CO2 removal, we bring the CO2 concentration back down to normal. Potentially we could overshoot, but we have really fixed this situation of low O2 and high CO2. So this is going to turn off the vasodilation and these changes to resistance and blood flow. So it's going to return us to our normal state. We know that that's a description of negative feedback, right? When we have a change away from a set point, and then we get pulled back down to our set point, that's negative feedback, turns off this loop. So this is still, all just a reminder, this is still intrinsic control of our blood flow because all this is local stuff right next to our arterioles. We haven't done anything with our nerves, haven't done anything with our brain. We're not sending signals from anywhere else. It's all local, all directly related to the arterioles right there. We have one other factor we want to think about for our purposes when we're thinking about the intrinsic control of blood flow to our organs, which is something called the myogenic response. So on Wednesday, we sketched out a little picture of smooth muscle. And we talked about some of the properties of smooth muscle, right? Our single unit smooth muscle, we saw it just had like one nerve kind of over it, sympathetic nerve. We'll talk about that in a second. We saw it was all connected by gap junctions, so it responds as a single unit. That's why it's single unit smooth muscle. We talked about the fact that it has tone. So it's at a kind of sort of medium rate of, it's a medium kind of constriction, so it can either dilate relax, get bigger, or constrict, get smaller. It's not at either extreme normally. We also mentioned that it reacts to stretch in a specific way. So when it stretches out, it has this property where it wants to rebound. So that's the key to our myogenic response. So our myogenic response officially is a change in vascular resistance in response to the stretch of blood vessels in the absence of external factors. Right, so myo we remember is muscle. Okay, so what we have happening in the myogenic autoregulation, because right, we're talking about the smooth muscle of the blood vessels. Again, this is very local, right? This is just a property of the blood vessel itself. It's not a signal from somewhere else. So our muscles, our smooth muscle around the arteriole is auto-regulating, self-regulating the blood flow. And so what we have happen here is that when we have increased perfusion pressure, and remember the perfusion pressure is just that pressure gradient across our organ, right? The pressure of the blood coming to it in the arteries minus the pressure of the veins taking blood away. So that's our perfusion pressure. So if we have an increase in perfusion pressure, we're going to increase blood flow because that's the delta P part of our equation, right? Flow equals delta P over R. So we're increasing the blood flow when we have an increase in perfusion pressure. But what this also does is it increases the pressure in the arterioles, right? When we have more blood flow, right, we're going to have more blood pushing out on the walls of the arteriole themselves, right? If you push a fluid really hard through a tube, causes a little stretch, causes a little pressure on those walls. So this increased pressure in the arteriole stretches the walls of the arteriole. This is just like when we were talking about the aorta, right? We talked about how when the heart beats, when it constricts, when it contracts, it sends blood into the aorta, which stretches and stores that pressure. So what's happening in the arterioles here is analogous. The increased pressure is stretching the walls of the arteriole. What this does in the arterioles, though, is it's stretching the vascular smooth muscle. So that's a little different than what happened in the aorta. In the aorta, we were talking about the elastic properties of the wall. Here, we're talking about the smooth muscle wrapping around the arteriole. That itself is reacting to the stretch. So the stretch of the vascular smooth muscle has a rebound, right? So we stretch out the muscle, doesn't like that, contracts back. Which as an analogy, you can kind of think of our reflexes that we looked at in phys lab, right? Muscles, when they get stretched, like to contract back, right? They have a set length that they want to be. So this stretch reflex is an inherent property of smooth muscle, this desire of the smooth muscle to go back to its initial state of tone. So we can think of this as vasoconstriction, right? It got stretched a little bit, and now it's constricting back. So we're shrinking down the radius when we have this constriction back, which decreases the blood flow. And the goal of this all is to keep the blood flow constant, right? Because if we didn't have this, I want you to imagine, right? So we have increased pressure, increased blood flow. It's pushing on those arterial walls. And if we just let it do that, if we just let it stretch out the arterial walls, let it stretch out that smooth muscle, that's basically vasodilation, right? So if we dilated the arteriolar walls, we'd then increase the blood flow because of the change to the resistance, right? It would lower our resistance and cause basically a positive feedback loop, right? That would increase blood flow even more. So we'd have this kind of runaway increase in blood flow. So that's why it's really important that we have this myogenic autoregulation, because we constrict that blood flow back, raising our resistance in order to make sure that we can keep that same steady flow of blood instead of constantly increasing and increasing the blood flow to that tissue. So of course, we have a nice little flow chart for that as well. So we're going to start out with the perfusion pressure in the arteriole. we're going to say that we're starting with an increase in the perfusion pressure. So that's an increase in that difference in pressure across our organ system that we're looking at, across our network of blood vessels. So when we increase the perfusion pressure, that's going to be due to an increase in flow, potentially. It's going to increase the flow through the arteriole, which means it's going to be stretching the arteriole or smooth muscle, right? So we have more flow stretching out the walls of that blood vessel. And then our smooth muscle bounces back, increases its constriction. Increasing constriction, right, lowers the radius. Lowering the radius raises the resistance. Raising the resistance lowers the flow. Then we have a negative feedback loop here, right, because we've lowered the flow. Right? We've lowered the flow, which means we're going to be lowering the stretch on the arteriole or smooth muscle, which means we can then lower the amount of constriction. So go back to our kind of set point and so on. So this is also a negative feedback loop. We had a change, we had an increase in flow, negative feedback loop bringing us back to our initial steady state. Okay. Questions about any of this intrinsic control of blood flow stuff before we move on to our extrinsic control? Okay. So one of the things that we're going to see, that our big picture here, why we're thinking about these arterioles going to specific tissues and to specific organ systems, is that we're interested in the relative distribution of blood to different parts of our body under different situations. So here we can see that at rest, our blood flow, right, is spread out between our skeletal muscle, our kidneys, our GI tract, our brain, and others, right? We have our heart here, we have our skin here. When we exercise, for example, right, we have active hyperemia, right? Our tissues that are really active in exercise, skeletal muscle, right? So our skeletal muscle is going to be dilating those arterioles, right? So it's going to get more and more blood flow. The rate to the skeletal muscle increases hugely, right? We're going from 1 liter per minute to 20 liters per minute. So then in these conditions, when we're exercising, skeletal muscle is going to be getting the bulk of our blood flow, the bulk of our cardiac output. We can see the cardiac output also goes up under exercise, right? And part of that is so that we have enough blood to feed to our skeletal muscle while maintaining blood pressure. So we can see that these are independently regulated, partially due to these local controls, right? These changes directly in the tissue and directly to the arterioles mean that what happens to the skeletal muscle can be separate than what's happening to the kidneys. We're going to talk about a little more how we have these different effects in different organ systems as we move into our extrinsic control of resistance and arterioles. Okay. So now we're moving to extrinsic control, which means we're going to start thinking about our nervous system, basically, nerves and hormones. So extrinsic control of arterial radius means we're thinking about not little local factors right next to the arteriole, thinking about factors that are coming from somewhere distant. Okay. So we want to remember all our equations. So this flow equals delta P over R, right? We've been thinking about changes in flow to our different organs. And we also looked at how we can sub in variables that we can measure about a body, right? Because this is just a general relationship between how liquids flow through tubes. But when we're looking at a body, we're looking at specific physiologic measurements. So we have our cardiac output is really our flow. Our mean arterial pressure is really our difference in pressure. The reason for that is that the central venous pressure is almost 0. So we can just kind of wave our hands a little bit there. It's a great approximation. The central venous pressure isn't really going to change anyway. So the changes to MAP are what are going to change the flow rate, divided by our total peripheral resistance, which remember is the resistance of all our blood vessels in the systemic circuit added up. But really, most of that resistance is coming from the arterioles. Right? We're controlling the arterial radius in order to control TPR. I think we saw that it was about 60% of that TPR is coming from the arterioles. So we can really make big changes if we change arterial radius broadly. Okay. I'm gonna switch our thinking a little bit as we move forward from now. Thus far, we've really been considering how changes in pressure and especially changes in resistance change flow. But as we move forward through the rest of the unit, we want to think of a kind of rearrangement of this equation. And you don't need to memorize a new equation, right? Because this is just the same equation, but we've moved stuff around, right? And what we're going to be interested in is changes to blood pressure as a result of resistance and as a result of changes to the heart rate, largely. So we can reorganize this equation, CO equals MAP divided by TPR, right? To get out of this, we just take the TPR from the bottom here on this side, move it over to the left, multiply by CO, and that gives us our new equation, which is MAP, which is fundamentally a measurement of our blood pressure, is equivalent to the cardiac output by our total peripheral resistance. Okay. So now we're going to see that any changes to our cardiac output, whether it's heart rate or stroke volume, are going to have an effect on our blood pressure. And any changes to our resistance are also going to have an effect on that blood pressure. So later we'll expand this out more and we'll see that MAP is equivalent to our heart rate by our stroke volume, because heart rate by stroke volume is our equation for cardiac output. This is why your extra credit was to go back over chapter 13, multiplied by TPR. Okay. So our mean arterial pressure is going to be dependent on our total peripheral resistance. And our total peripheral resistance is dependent on the radius of those arterioles. This is just my reminder to do a poll question. That's good. Okay, so now that we're thinking about extrinsic factors, I want to think a little bit more about our nervous system, specifically our autonomic nervous system, specifically our sympathetic nervous system. Okay, take another second. I'm going to end the polling. Okay. So our question was, which of the following is the main neurotransmitter produced by the postganglionic cells of the sympathetic nervous system, which is one of the two branches of our autonomic system? The answer is going to be norepinephrine. And so we're going to take a look at that right now. So when we're talking about our sympathetic nervous system, this is a picture of 3 pathways for our sympathetic nervous system. We have a preganglionic neuron coming out from our central nervous system, which is our brain and spinal cord. Then it's going to have a synapse here with our postganglionic neuron, right? So the preganglionic neuron is the one that releases acetylcholine. It's cholinergic. Our postganglionic neuron we refer to as adrenergic because it's releasing norepinephrine. We'll also see an association with epinephrine, which can also be called noradrenaline or adrenaline, which is where that term adrenergic comes from. Okay, so our sympathetic nervous system, from the perspective of our tissues out in the body, is really releasing norepinephrine. That's its main job from our effector organs perspective. When we talked about this way back in our third unit, I believe, in ANP1, we also looked briefly at different types of adrenergic receptors. So the proteins on the cells in our effector organs, those tissues that are releasing or that are receiving the norepinephrine. Okay. And we talked about the fact that we have alpha receptors and beta receptors, so two different classes of adrenergic receptors. And then we broke that down into alpha 1, alpha 2, and beta 1 through 3. So we're going to be thinking about these again now. Specifically, we're going to be thinking about our alpha receptors versus our beta 2 receptors, which is why I put this chart back down here. And when it becomes relevant in the rest of these slides, I've pulled it up again. But go ahead and pull this up in your book later. I think it's not in your copy of the slides, but we've seen this before, so it should be somewhere in your notes. Before we were focusing on their effects, their excitatory versus inhibitory effects, and their relationship to cyclic AMP. But what we're going to be focusing on now, as we're thinking about our arterioles and our cardiovascular system, we're really going to be focusing on this third column, this relative affinity for norepinephrine versus epinephrine, and also the type of organ affected. So we kind of waved our hands potentially a little bit over here, but we're specifically going to be looking at the distribution of these different receptors in the vasculature, right? The vascular smooth muscle versus our effector organs in, for example, our blood vessels in the skeletal muscle and the cardiac muscle. We'll find out. That's what it means by some. Okay. Just make sure. Okay. So our extrinsic control of arterial radius and therefore of our mean arterial pressure, right? Because we now know that the relationship we're interested in there is, I'll write it out again, right? Our mean arterial pressure is regulated by our cardiac output and by our total peripheral resistance. All right. Okay. So both of these are going to be regulated externally by these chemicals we have coming out of our nerves, right? So these extrinsic mechanisms are going to be some direct sympathetic activity. So directly that norepinephrine being released by the sympathetic nerves, and also some hormones, which we'll talk about. Okay. So when we're thinking about the sympathetic control of the arteriolar radius, we're thinking about, right, that sympathetic nerve, basically, that's lying on top of our bed of smooth muscle around the arterioles. So we have sympathetic innervation to that smooth muscle coming out of our autonomic nervous system. So when that sympathetic nerve releases norepinephrine, it acts on the smooth muscle surrounding the arteriole, and that's how it's going to control the radius, right? So this is why it's extrinsic. It's a signal coming from far away, right? Ultimately coming from your central nervous system. The smooth muscle of most arterioles, but not those in the brain, because we want to make sure that our blood flow to the brain really doesn't get messed with, has alpha adrenergic receptors. Okay. So, thought I popped that on there. I'll be on the next slide. Okay. So most arterioles have alpha adrenergic receptors, which if we flip back, right, our alpha adrenergic receptors, they like norepinephrine a lot. They like epinephrine a little bit. So norepinephrine does bind to those alpha adrenergic receptors. They really like norepinephrine. And the response in the smooth muscle when norepinephrine binds to an alpha adrenergic receptor is vasoconstriction. So if you have smooth muscle with a alpha adrenergic receptor, when norepinephrine gets released by the sympathetic nerve, it's going to come into contact with that alpha adrenergic receptor. It's going to cause that smooth muscle to contract, which means it's going to constrict the arteriole. When we constrict the arteriole, that increases our resistance, right? And because of our new understanding of the relationship between mean arterial pressure and total peripheral resistance, right? So if we constrict and we raise TPR, right, increasing our resistance because we've constricted the blood vessel, this has a direct influence on the mean arterial pressure, right? So this is a proportional response, a raise in blood pressure, right? Okay. It gets more complicated when we think about the arterioles that go specifically, I'm going to even underline them, right, specifically to the skeletal and cardiac muscle, right? So most of our blood vessels have those alpha receptors, but our skeletal and cardiac muscle, we're going to have a different distribution of adrenergic receptors around the arterioles there, right? So we're still thinking about arterioles, but specifically ones in your skeletal muscle and cardiac muscle. And here we're going to see that instead of having alpha receptors, we're going to see we have beta-2 adrenergic receptors. So here, it's going to become important, the difference in the affinity of our alpha receptors and our beta-2 receptors for epinephrine specifically, but also norepinephrine. Okay, so we can see that our beta-2 receptors are particularly, right, we have two of these little carrots, very responsive to epinephrine, although they will respond a little bit to norepinephrine. So that's the thing we want to keep in mind as we talk about these next couple of slides. Right. So when norepinephrine binds to alpha receptors, it causes vasoconstrictions, so the arterioles get smaller, blood pressure goes up. Epinephrine can bind to either alpha receptors or beta-2 receptors. So we got alpha receptors everywhere. We got beta-2 receptors and alpha receptors in our skeletal and cardiac muscle, but mostly beta-2 receptors. And epinephrine has a different effect depending on whether it's bound to an alpha receptor or to a beta-2 receptor. That's why we have all these different receptor types, right? They can really vary in their specific effects in different tissue types. So this table isn't in the slides for this lecture. I just pulled it up from one of our previous lectures on the autonomic nervous system because I think it's helpful to consult. And I can find specifically what chapter that was in for you guys in the textbook. Okay. So epinephrine is going to be binding alpha receptors and causing vasoconstriction. But when epinephrine binds a beta-2 receptor, it's going to cause vasodilation. Someone mentioned that it's page 311 in the text, this chart that I have up. So it causes vasoconstriction at alpha receptors, vasodilation at beta-2 receptors. And the other thing we note is that it has greater affinity for those beta-2 receptors. So we're going to see that the effects of epinephrine are going to be different depending on how much epinephrine we have. Because if we have a little epinephrine, it's going to get hogged by beta-2 receptors. But if we have a lot, we're going to see that it's going to affect both types of receptors because we have extra and we have enough to basically wash out this effect of norepinephrine. So this is really the fundamental background of these next couple slides. So the effects of epinephrine on the arteriolar radius are concentration dependent. At low concentration, it binds beta-2 receptors because of this relative affinity, right? Beta-2 receptors are actually our only receptor that really, really loves epinephrine. So if you have only a little bit of epinephrine, it's all going to get sucked up by those beta-2 receptors. The effect on beta-2 receptors is vasodilation. So if we only have a little bit of epinephrine, overall we're going to see vasodilation. We do want to remember that beta-2 receptors are not just spread out everywhere, specifically talking about the heart and skeletal muscle. Okay, so we're going to have vasodilation going to the heart and going to the skeletal muscle. Now, as we start to have higher concentrations of epinephrine, right, your adrenal gland producing lots and lots of epinephrine, right, we're going to first bind those beta-2 receptors as we're ramping up. But then once we have enough, we're going to have spillover, right? Those beta-2 receptors may all be occupied. So the epinephrine is free to go bind to other adrenergic receptors. So it's going to start binding alpha receptors. We have the opposite effect on arteriolar radius when we're bound to an alpha receptor versus a beta-2 receptor. So we're going to see the vasodilation happening in skeletal and cardiac muscle due to their high proportion of beta-2 receptors. Those beta-2 receptors are vasodilating. So in the skeletal and cardiac muscle, we're going to see a decrease in our resistance, which would be a decrease in blood pressure. So that would be our effect at initially the low concentration, and then that's going to be the additive effect even at higher concentrations of the skeletal and cardiac muscle. But when we're at higher concentrations, we're also binding those alpha receptors. Alpha receptors are doing vasoconstriction. So this is what's happening in most vascular beds. So the other arterioles, not in the skeletal muscle and the heart. And as we have vasoconstriction, we're going to maintain or even increase the total peripheral resistance, right? Those blood vessels are shrinking, so their resistance is going up. So it's going to counteract the vasodilation that's going on in the heart and in the skeletal muscle. So this is going to have the effect of maintaining blood pressure because basically, right, like we have our equation, our mean arterial pressure, our cardiac output, and now we're thinking about changes to total peripheral resistance. Okay. And quick note, as I do these drawings, if you happen to be colorblind or otherwise have difficulty interpreting the colors I'm using on my screen, please let me know. I try to pick well, but if it's not working, I want to know. Okay, so our effects on total peripheral resistance here, when we have lots of epinephrine, we're decreasing TPR with our skeletal and cardiac muscle. Okay. Okay. We're vasodilating and decreasing TPR, but we're making up for it with our vasoconstriction everywhere else. So these basically cancel each other out, maintaining the blood pressure. Okay. We'll see that sometimes, and even maybe usually, the vasoconstriction from the vascular beds exceeds the vasodilation in the skeletal and cardiac muscle. So the dominant total effect is vasoconstriction. I'm getting a question about the vasoconstriction during fight or flight response. And yeah, that's exactly what we're talking about here. So when we have the fight or flight response, we have sympathetic innervation going to our adrenal glands, which release epinephrine. And then we have this kind of balance happening between what's going on in our skeletal system and the cardiac muscle, which are preparing us to like run away or to fight, versus what's happening in the other vascular beds for like, you know, digestion or whatever. So when we said that in this fight or flight response, we increase our activity of the skeletal and cardiac muscle, and we decrease our activity in like the GI system, that's part of what's going on here. So it's this balancing act, because we want to keep blood pressure, because if you drop down your blood pressure, I don't know if this happens to any of you, right? But like if you're sitting and then you stand up, suddenly sometimes you have a head rush as your blood pressure drops, right? You wouldn't want that during a fight or flight response, right? Like that guy we saw on the first day, right? He had issues with his blood pressure when he stands. You wouldn't want that happening during a fight or flight response. So it's important that we at least maintain the blood pressure in order to do this fight or flight response. Now, overall, we do end up raising our blood pressure a little bit usually, depending on the strength of this response. But yeah, this vasodilation in the skeletal and cardiac muscle kind of counteracts that. Right? That's why we have both going on at once. Okay. So our cardiac output, we saw increase during exercise, right? So that was this diagram at the bottom here. We kind of looked at how independent control of our different organ systems can change the distribution of blood, but it would also overall change how much blood is being pumped out by your heart, right? If your skeletal muscle suddenly super dilated, right? It's getting a lot of blood. That would also have effect on the total cardiac output because it has an effect on the blood pressure. And this is dependent on those receptor types. So the dilation of blood vessels in the skeletal muscle and heart are increasing. Right, we have vasodilation happening because of those beta-2 receptors, right? So that's why this chart with our skeletal muscle, we have so much blood flow to the skeletal muscle. So that's kind of an additive effect right there with our active hyperemia, right? In the skeletal muscle, we have super active tissue and our hormones are vasodilating there, right? So those two effects would add together. Then we have constriction of vessels in the GI tract and the kidneys because you don't need to be digesting food or really filtering your blood too much during exercise or during a flight or flight response. So those alpha receptors are constricting. So this is creating a disproportionate flow of blood, mostly to the muscles. So finally, this last little bit, we do have some hormones that are also extrinsically controlling our arteriolar radius. So epinephrine counts as a hormone as well, and this is what we've just been talking about. We have two more. We have vasopressin, ADH. We have angiotensin 2. We'll talk about these a lot more when we get to the renal system. These are going to be very important when we talk about maintenance of blood volume through the action of the kidneys. But a very, very short, brief explanation of this is that vasopressin is secreted by the posterior pituitary. So that was the part of the pituitary we didn't talk all that much about in ANP1. So posterior pituitary was vasopressin and oxytocin. Its usual role is to increase water reabsorption by the kidneys, but it also does vasoconstriction. So it's going to be raising blood pressure by vasoconstricting. Vasoconstricting, right, and changing the resistance and therefore changing the blood pressure. Angiotensin 2 has the same effect. We'll also be talking about the angiotensin-renin system when we get to the kidneys. But in the blood vessels, it also creates vasoconstriction, increasing total peripheral resistance, increasing blood pressure. So those are two other hormones that come into play here. Okay, and that is our time, and that is the end of this packet. So I will stick around for questions, but I'm going to stop our share and stop our video now.